I'm the founder of Retreat and Learn. Thank you very much for joining us today. Today, we have Forrest Bowling, who is a doctoral candidate in biochemistry at Stony Brook University, and he was recently published in Nature Chemical Biology. I got to tell you, I'm really excited for today's interview in a way that I'm not normally, because Forrest also happens to be my son. So if you are live with us now, please say hi. I know this is nerve-wracking for every single person I interview, and they like to know who's here and know that there's a friendly face. So please say hi in the comments, and we'd love to know that you're here. As we talk today, please post your questions in the comments, and that way we can answer them while we are live. And if you want to easily find future talks or past topics, you need to follow us on Facebook or subscribe to our YouTube channel. And then you can also just go to retreatandlearn.com and sign up for our email list. And all those different ways are going to make sure that you know the next interviews that are coming up. And we have some really neat interviews coming up. So with that said, I'm going to bring in Forrest. So welcome, Forrest.
Hi, Mom.
Hi there. This is a fun interview. It's kind of an interesting one. I've interviewed a lot of people. I've not been able to interview my son. And I'm not just interviewing Forrest because, he's my son. It's also, he has some really interesting things to say for people who are thinking about going into the science careers or that just don't really know what a scientific researcher does. So Forrest, if you could just share that, what do you do every day?
Yeah, that's actually it's a good question and a complicated answer.
I bet.
So currently a graduate student at Stony Brook University. And so when I tell people that, they think I'm just going to classes or taking tests all day. But really at this point, I'm just doing research, like a 9-to-5 job almost. The first 2 years of when I started at the graduate school, it was a lot of taking classes, taking tests. It looked like school with doing some research, probably 20 hours a week. But then after my second year, I then proposed a thesis project for the rest of my time there. And then I had a committee approve it. And then I transitioned into full-time research. So I'm in the lab any chance I can get. And I start the day proposing experiments, designing them, looking at yesterday's experiments, and then spend the majority of the day actually being in the lab, running experiments. And then at the end of the day, doing statistical analysis on that, or preparing presentations, or writing it up in like a readable format. So it's a very varied day of a scientist, at least as a graduate student.
All right, and that was something I didn't understand when you first went to Stony Brook. I just didn't understand. For me, I thought that somebody who was getting their doctorate was going to sit in class for the next 5 years, just like an undergrad, before they got their degree. So it was very surprising to me that that's how it happened. That you actually started off in class, but then you actually transitioned to almost like a real job that you went to every day. Is that typical for anybody who's in the doctoral program, or is that just at your school or in biochemistry?
It'll depend on the school and program. So when I first started, I took the classes and then did like rotations where, because you don't know what type of research you even like yet or what professors you're going to get along with. So I do different rotations with 3 different professors for about 3 months each, like trying out, oh, do I like this type of science? Oh, I don't really get along with that guy. So then you find the good spot for you to do like your entire thesis research. But even at Stony Brook, that differs what type of scientists or even like what graduate degree you're going after, such as like chemistry, you could potentially go straight into your research. You might just know directly which professor you want to work with. I don't really know about like liberal arts PhDs because I do wet lab work all day, but they also are doing just doing a ton of research on their own and all types of stuff. Yeah.
All right. Now, you just said a word that I don't understand that probably a lot of scientists do, but what is wet lab?
Wet lab is kind of how we differentiate between working on a computer, working like in an office type setting, or pretty much anytime you have your gloves on. So if I'm working with like insect cells or like chemistry, that's like a wet lab setting. Dry lab, you're either doing computational models or doing just pure research, like literature research. Yeah.
Okay. So like a simulation might be, do they call it dry lab or is it just not wet lab?
Yeah, I would say a dry lab.
Okay.
Yeah.
Okay. So a simulation or doing research, something that's more in a literature type setting or a computer type setting. would be. Okay, got it. Okay, I've never heard that term, so that's pretty interesting.
Okay.
I know it's a very common term to you, but probably not to a lot of people watching. So now that you're into the part, like you're starting your last year or potentially your last year, you said you go into the lab each day and you're doing your research. How do you decide? I mean, do you have a professor that works alongside you? Who tells you what to do? I mean, because you're not a full-fledged doctor at this point or a researcher. So who helps you figure out what to do and guides the work that you're doing?
Yeah, that's a good question. So I'm in the lab of Dr. Michael Arola. So he's kind of like my PI or principal investigator on like the total project. And so all projects in his lab kind of fit in a certain like theme. So we focus on like how different fats in your body get broken down into different types of fats. And so I'll talk to him probably about once a week, try to like propose certain experiments to him because he has this like vast amount of knowledge and wisdom because he's been working with biochemistry for much longer than I have. And so any mistakes I might have when proposing something or maybe a tip he's had like picked up over the years, he can let me know. And so then once I talk with him, I usually then do it pretty much independently the rest of the week or even a few weeks after that. And so I'll just I'm in the lab. And so I'm making the decisions by myself. If I need help, I'll ask either other graduate students. We have other research scientists in the lab who already have their PhD. We have postdocs in the lab who I can then ask. It's a very like collaborative environment. So you're working not only with like your boss, the PI, but also with other students and other workers.
Okay. I forgot one question I meant to ask at the beginning. So when you were still an undergrad and you went to UMBC and so you were, looking at doctoral programs and deciding to go, into one, what were the steps to actually get into a doctoral program from undergrad to the grad school?
Yeah. So it's pretty much similar to applying to an undergraduate degree. The only difference is you'll need, instead of an SAT or ACT test, you usually take this test called the GRE, which I don't remember what it stands for, but it's another standardized test. And then you'll get letters of recommendation, which are more important than I think than the GRE even test itself. And then you start applying to schools. And so I only applied to 3 total schools, just schools I knew certain professors there I wanted to learn from, or certain types of biochemistry that they're like really good at that I wanted to learn from. And then you hear back from them, and then you actually go and do interviews with several different professors at that school. And so you go as a big group of around, there was 20 undergraduates there, and we all just do these all day long interviews and tours of the campus. And then you hear a letter back in a few weeks or so, and then you're into graduate school.
Okay, no, that's pretty good. So is it very competitive? I mean, is it very difficult to get into to grad school in the biochemistry field?
It's all depending on the school. It's pretty much similar to undergraduate, where there's definitely easier schools to get into that will take most people, because graduate students are not only students, but they're also kind of sources of cheap labor for the universities.
Right.
I not only do all the lab work, or most of the lab work as a graduate student, but also I've taught classes at the graduate school, involved with certain things. So schools definitely want graduate students. It's kind of like a lifeblood of universities. But then also other schools are much harder to get into. Shouldn't be a shock. It's pretty hard to get into Harvard for a graduate school. I got denied from one school on my applications, and I think I had a pretty good application. So.
Okay. No, that's all pretty interesting things. And when you went to school, it was very interesting to me because I just didn't understand how that all worked. I mean, I stopped at undergrad, so it all seems very commonplace to you. It doesn't seem commonplace to everybody. So we did actually have a question. We have a question from Jennifer. She wants to know what your PhD thesis is. What's the topic?
Yeah. So I'm getting my doctorate degree in structural biology and biochemistry. And structural biology is kind of looking at the sub-microscopic structures in your body. And we try to look at what they look like. We mainly focus on proteins. And so proteins are kind of like the tools of the human body. For example, hemoglobin. Hemoglobin is this protein that carries oxygen throughout the body. It's this kind of tool that the body uses. And we use a variety of different instruments to determine the structures. Personally, I use X-ray crystallography. And so we're using X-ray crystallography to look at the structure of this one protein involved with how your cells grow, how they generate new blood vessels. And it's also this like really fantastic like cancer therapy target. And so when you determine the structure of a protein, you can then determine certain medicines. You can design them to fit into that protein, which can change their activity, leading to hopefully with our protein reductions in tumor size and growth.
I have so many questions now based off of that, what you just said. So first, and I'm probably not going to say it correctly, but what is X-ray? Give me the word.
Crystallography.
Yes, thank you. So what is that?
Yep. So proteins are super small. They're like, if you look at your thumbnail, it's around 1/10,000,000th smaller than your thumbnail. They're smaller than a microscope can see. So you need to use like different types of tricks. And so I'm sure everyone made like rock salt as kids using just sugar, or you've seen like salt water dry up and you see these big salt crystals show up. Certain proteins actually make crystals in the exact same way. They'll make these like really beautiful crystals. If you want to just like Google search protein crystals at some point, like they make all types of different like shapes, like football shapes, long rods. It's like, I like crystallography because it's kind of like a witchcraft of science. And so once you get this crystal, you can then take it to, we blast it with X-rays, and we do this at usually synchrotrons or X-ray beam sources. We are really lucky at Long Island because we have Brookhaven National Laboratory really close, and it has this gigantic synchrotron, which is like this particle accelerator.
All right, well, slow down. What's a synchrotron and what's a particle accelerator?
A synchrotron
I know
generates
I'm asking tough questions for me, but
I still barely understand. I still barely understand what a synchrotron is. I love going there. It's kind of like this gigantic circle that you're able to generate very specific types of light with by using these gigantic magnets. And by like a really big circle, I'm talking about like, it's so big to get around it, they have bikes all around the campus to get around this gigantic circle. So we use that to generate very specific types of X-ray light.
So the most amazing thing about that is that you just talked about how small that protein is, that it's so small that it can't be seen by a microscope, and you need something to see it that is so big that you need to drive a bicycle around.
Yes.
And that's amazing to me, and it's something that I think most people, I didn't know that, so that's pretty interesting. So pretty cool things. So the other part of it is that you said that the research that you're doing, hopefully will slow, that you can, once you discover it, you might be able to figure out how you can slow the protein growth that then will eventually also change the tumor growth, because the two of them are linked together. Is that correct? Did I say that correctly?
Yeah, so the activity of my protein. So once it does its like certain effect, it leads to increased like cell growth, and tumors are just made of like up of your own cells. And so I actually have a picture if you want me to share screen.
Yep, I sure can.
So are you able to see my screen right now?
I can. We can all see this.
Yep. So on the left, that's like the structure I was able to determine with X-ray crystallography. It looks like a bunch of ribbons and arrows, but that's just kind of like a cartoon representation of like the electron density of this protein. But on the right is some of this like work by some of our collaborators here at Stony Brook, where they inserted tumors into these mice. And so on the left, you see a normal mice or wild type WT. But then once you take the mice and you knock out my protein. So PLD1 is the protein I study. If you take that protein out of the DNA of these mice, they're relatively healthy, but you see this drastic reduction in tumor size. And so if we can design a drug that's able to turn off my protein, similar to how they took it out of these mice, we could potentially see this like also strong reduction in tumor size for like as a great like cancer therapy target.
So if I understand this, how did they take maybe this is probably a little beyond this, but they actually removed the protein from the mice?
Yeah, and so it's actually like funny sometimes, like I'm like a biochemist, but like how little biochemistry I actually don't know. And so like I know a lot about my own protein, I know a lot about how it works, but how mice models work or how like anything pretty much larger than a cell, I actually don't know how they do that. But they are able to like turn off this like protein in their body and like grow them up. Yeah.
Huh, that's cool. But they're not, that wouldn't be the goal within a human translation. They would actually just want to turn it off so that the protein wouldn't grow.
Yeah, so you'd want to
try to remove them.
Yeah, so the main focus right now would be to like design a drug. And so that drug you could like then take into your body, and they would be able to specifically interact with that protein like essentially turning it off, like the same as like a light switch almost.
Okay. So that kind of leads me into my next question in terms of, and this isn't really your field. I understand that you're studying a very specific, and that's one of the things that I've learned from, you being, going through this process is that science is very, very specific. You study a very small, little specific area of science, and you aren't really linked to some of the other things. But one of the things that is on everybody's mind is vaccines, in terms of, vaccine development. And while it's not your field, I was wondering if you could shed some light on just kind of the process of how vaccines are developed and, why we can't get a vaccine done tomorrow.
Yeah. So vaccines are basically going back to like how your bodies react to a virus. So you'll get sick with the virus, and then eventually your body kind of like engulfs the virus and then breaks it down and like displays certain parts of this virus, and that'll help it generate the antibodies to like then if you get infected again by a virus, it then has like almost like a wanted poster of like, oh, that's the bad guy. Now we know what it looks like, and then it can swarm that and destroy it before it gets you sick again. Vaccines are kind of the exact same way. They are able to take parts of the virus they can put it into your body, but it doesn't have the replication of a virus, so it can't make more of itself. It won't be able to make you sick. It's just a small fragment that your body can then recognize. So say you're only putting in, instead of the whole bad guy, you're putting in just like its hand, but its hand is very specific. And so then once you put the hand back, if it sees the hand again, it has the wanted poster, it knows, oh, that's the bad guy. So you're always safe from like the virus unless it mutates from then on out. And so vaccines work in that way is we can take just a protein very specific to, like, say, the coronavirus, or the DNA or the RNA of the protein very specific to the virus. And then the difficulty is mass producing this. And then also, I think, making sure it's like safe for people. Like, again, this really isn't my field, but that seems to be the biggest tough is there's so many different ways to look at making vaccines. And so they're just pushing it every single way.
Right. And it takes time. I mean, all these things, the science is a very slow, steady, methodical process. So it does take time. And so I mean, I know this is not your field, so I appreciate you, at least, doing your best to answer that question. And one of the other things that you and I have talked about too is that right now, because people are scared, and so there's a lot of things being passed around on the internet. Somebody telling their brother, "Hey, this is what I heard," and so there's a lot of misinformation, about COVID-19 and just how viruses are spread. Is it on surface? Is it not on surfaces? Does it last 5 days? And so all this information is being spread. Do you have anything that you want to just discuss about that in terms of, how do people make sure that the information they're getting is accurate?
Yeah, it's hard, because misinformation travels like a virus. It just spreads and is very shareable throughout social media. And then before you know it, it's everywhere. And then it's affecting how you're making decisions in your life, how you're thinking about decisions made by like the government. And so when you see a posting on like social media or like a headline, like think about it, like, is it a very like clickbaity title? Like, it's like, oh, what are they hiding about the coronavirus from us? Or is it a more, when you see that title, I would like read the article, see what it looks like, and then go to a different source, like a more respectable source, like the BBC or like Nature, or like a more like source you're going to find like good science at, and see like, okay, how do these compare? What did all these like read up or like summaries of scientific findings, they should be sourcing to the actual paper that the scientific discovery is based on. Okay. And so you can go to that, and there should be an abstract. And so you can like then look at the abstract. They're usually written in more general terms for like a general audience. And you can say, okay, did the headline of that really represent what the scientists were trying to propose in this experiment? Like, are they like trying to like make it sound bigger than it is? Are they misinterpreting certain aspects of it? So I think just finding multiple sources of information, and then also like looking at them together and like be rational. Like, is it also, don't look for just headlines that are confirming with your pre-existing beliefs. Make sure you find like everything from respected sources, and so you can get like a really good knowledge of the situation.
And also, yeah, I guess before you spread it, double check that, not just for your own source.
Yeah. Know the power of your sharing, because people are going to see if like their cousin's sharing something, they know them, and so they respect their opinion in some way. So know the power of sharing on social media.
Yeah, well, I appreciate that, Forrest. I mean, I think that it's something that we could probably do for not just having to do with COVID-19 and viruses, but probably all information on the internet. Check your sources. It's probably very good information. I'm going to kind of go back to your schooling because I think some of the people watching today really want to know a little bit about like the process and things. So once you go into the program, is there a set time frame for you to graduate and finish?
No, that's why PhDs, it's not the same as undergrad where it's like, oh, you usually average in like 4 years. PhDs are just whenever you're done. So whenever your committee, which are usually organized of 3 professors that are like not your PI, you meet with them once a year. You'll say like, okay, here's the research I've done so far. And they kind of decide like when you're ready to defend, like when your thesis has reached like a good like endpoint. And when that benchmark is depends on your committee, depends on the programs you're in requirements. So mine requires me to have at least 1 publication in a journal before I'm able to graduate. Some might require 2, some might not require any at all. So you'll hear PhDs varying from 3 to like 8 years, which is like scary. So it just depends just how lucky you are with science, because science can be unforgiving. You can get like a really good project and you're like, oh, this has got to work. And then you just get negative results after negative results after negative results. And you're just like, it's not your fault. It's just Science is mean. It's really mean sometimes.
Yeah. Yeah. That's got to be disappointing. You work really hard on something. It doesn't mean that you're not doing a good job, but sometimes it just doesn't pan out. I think we even all experienced that back when you were doing your 3rd grade projects. Sometimes you go to your science project, it didn't work out. And sometimes that's the conclusion. So how long are you in? I know the answer to this, but how long are you in your program right now?
So I'm in my 4th year currently.
Okay.
And so my fingers crossed expected graduation date would be 2021. Haven't quite pitched that to a committee, but I've heard rumblings they're okay with that. So that's the hope.
All right, we'll see. I mean, and again, you don't know until you're done when you're done. So after you graduate, what are the career choices after you graduate with a PhD in biochemistry? Where can you go with that?
Yeah, there's a lot more than I thought going into my PhD, because you originally go in and you always hear 2 things as a PhD student, like, oh, are you going to be a professor or are you going to do research in like a biotech company?
Okay.
Like those are just like the 2 career paths you hear constantly. But then during my time at Stony Brook, we were able to take classes. We have this Center for Communicating Science here at Stony Brook, which actually was founded by Alan Alda, the actor. So that opened up some certain avenues of career choices to me. But then also I've been become the president of the Graduate Career Association here at Stony Brook, which has also shown more paths. And so not only are those 2 paths available, but you can do government research. You could also be an editor in one of those journals we're publishing at. You could be a medical science liaison, which works with doctors and also the technical companies as kind of this in-between of scientific knowledge that's representing the hospital. You could even go into data science. It's rapidly expanding field because scientists are really good at taking a large amount of data and interpreting trends and making predictions from that. You could even do, I know as consulting, that's another big thing. Consulting for all types of PhDs is another avenue of careers because you're presented with these really hard, complicated questions, which there isn't quite an obvious answer to. And so you sit down, you're able to break it down and find doable solutions to these problems. And so PhDs, it not only trains me in biochemistry, but it's also trained me in problem solving, teamwork with people from different backgrounds, and also distilling large amounts of information. I've read countless papers and distilling it into small PowerPoints or small blurbs, just trying to find these trends and conclusions. So there's a lot of avenues for PhDs that go outside of just research or wet work.
Yeah, I mean, it sounds like, right now the world is your oyster in terms of trying to figure this out. So that it probably is a little bit, it's great. It's also a little overwhelming, I'm sure, but it's great to have choices. So, all right. Well, is there anything that you want to add either about your project, about your schooling, or anything that you think people might want to know about, being a scientific researcher or being a doctoral candidate, any of those topics?
I endorse it. It's definitely a grind of a post-career choice. It might also seem easy for people to just oh, I'm in school, I'm just gonna keep doing school. But I really enjoyed it. I like grown a lot as a scientist and also as like a worker, like in like a team setting. So I think it's a really fantastic career opportunity that opens up a lot of doors that you wouldn't have available as just a bachelor holder. Yeah, that's about
Okay.
it.
Yeah, no, I mean, I've seen you really enjoy what it is that you do every day. And I know that it was a tough decision going, do you stop at bachelor's or do you keep going? And I know that was a choice that you had to make. But I've seen definitely that you enjoy every single thing that you do. So I think that that's really great advice that you're giving people who are thinking about going that path. If it's your passion, it's definitely a good place for you to be. So well, I appreciate you joining us today and answering the questions for people that wanted to know a little bit about that career path. I think it's something that was confusing to me, so I know it was confusing to other people too. As a reminder, if you do want to watch some of our other videos and interviews that we're going to be doing, you definitely need to follow us on Facebook. You need to subscribe to our YouTube channel. Next week, we have a really awesome interview on grief and how you can use writing to get over that grief. So if you want to get notifications of all those interviews, you need to join along. So thank you very much, Forrest. And I'm sure I will see you at some point when we're allowed to see each other once again and COVID is over.
Yeah, I'll find my way back down to Maryland soon.
Great. All right. Well, thanks so much. And we will see everybody soon. Bye.